Everybody. Okay, good afternoon, everybody. My name is Nubi Olubunmi, and I will be taking an important, equally important aspect of this summer school, which is the choice of sample containers. Sorry, I need to close some. Okay, yeah, choice of sample containers for marine sample collection. Yeah, in as much as it's a small component, but it's very vital in the project design when you are set to go to the field for your sample collection. You know, when you want to go for an expedition, you need to lay more emphasis on the sample container that you are taking to the field. So,
Nubi, I'm sorry to interrupt. Okay. We can see part of your screen, but we can't see the right-hand portion of your slides.
Oh.
I think it might be the Zoom that's causing the trouble. Oh, that's great. Perfect. Thank you.
Oh. So will it be okay if I run through the slides in this mode? It won't be in a presentation mode.
Yes, this looks fine to me.
Okay. I'm sorry about that. Yeah, maybe no some challenges. Okay, so yeah, the outline that I will follow, I will quickly talk briefly about sampling and then also different objectives. Then also the importance of sample container and then general considerations given to sample containers and also the fundamental guiding rule. The analytical protocol or protocols to be used for sample analysis often requires the use of a particular type of container. And then also the type of container also may depend on the sample matrix and your analyte of interest. I will explain later what analyte of interest, you know, for some of us that we are just, we just have little knowledge about, sampling or sampling techniques. So it's important that I define standard operating procedure, which is very, very key when a project is about to kickstart. So this is the guiding document that researchers must follow in order to execute a particular research project. So it's the standard operating, a standard operating procedure is a set of written instruction that describes the step-by-step process that must be taken to properly perform a routine activity. So this presentation that I'm making is not for you to look away. It's not for us to look away from adhering strictly to SOP, but rather to bring out some scientific details of its content in relation to sampling and sample containers. So, you know, as a scientist, you don't just follow SOP the way, I mean, just like that, but you need to understand, you know, the step-by-step content of that SOP. What are the scientific indication? What are the scientific explanation, you know, given to each of the steps that you need to follow? In the context of this presentation, I won't be looking generally into different, you know, aspects, but I will focus more on some of the unifying considerations, you know, yeah, because we have different different fields when it comes to marine sciences, and the considerations for sample containers vary, you know, based on that. So, but I will talk on, you know, some unifying, you know, considerations as to sampling and marine, sampling and the sample containers. So, to simply put, sample is a part of the whole that can be examined to see what the rest looks like. And then we have different oceanic samples. We have different types. We have the water itself. We have the biological samples. ranging from phytoplanktons to higher organisms. We have geological samples, talking about sediment, mineral deposits, and then also we have debris, in terms of plastics and other solid materials that you can find that you might be interested in, in sampling from the ocean. So sampling now, it is the method or the process of selecting a small section of a larger group in order to estimate the characteristics of the entire group. So as I've rightly defined, sample to be a portion, a small portion that you will need to study to characterize, to see how others look like, other components. So the process of establishing the collection of that sample is generally referred to as sampling or sampling method. So several reasons, we have in oceanography for making us to sample. So it's an important step in oceanography, and then it has a lot of reasons, to back it up. So motion parameters still requires it. So motion parameters still require sampling, basically because of the mobility of some equipment, or possibly the mobile types are, expensive.
Nubi.
I can hear you. Can you all hear me?
Yes, we can hear you, sir.
Okay, so I will go on. Thank you. So yeah, as I've said, due to the immobility of some equipment and maybe the mobile types of those equipment, they are expensive or even yet to be produced, or possibly due to the pretreatment that you need to give to the sample prior analysis. We call it prior analysis procedure. So you cannot just determine that a parameter on site. It has to be sampled and taken back to the laboratory. Also, analyzing every drop of water in the ocean is practically impossible. And even if it's going to be possible, it will be time consuming. And you know what they always say? Ship time is expensive. So sampling allows us better coverage within the shortest practical time. So let's look at sample design as a whole. The first component that I will mention here is the objective. The objective, of your project, the objective of your sampling is very key, is critical to sampling design. The objective of your study, do you want to do a pollution study? Do you want to carry out a productivity study? Or is it a climate change impact study? Your objective will go a long way to determine how effective the sampling design, will look like. And I've given a schematic diagram, which gives, all the components of sampling design. So the first one that I've mentioned, and that I've mentioned earlier, is the objectives. What are the parameters, that you will need to use to achieve your objectives? What is the level of accuracy? What do you want to put the data, how do you want to put the data, to use? So those are the questions that, you need to ask. And all these are achievable onshore, right there in your laboratory. You need to be able to look into that. They are all achievable. And you can even improve and modify them even during the cruise. As you are going, you can look at it again. So all these are flexible and, they are achievable at any given point. So you need to be able to establish your sampling location. Where do you want to actually sample? Where can you get the best in terms of representation of sample? You need to be able to establish that. What is the sampling frequency? So, but when it gets to a point whereby you are on the field and you need to collect your sample, that is the green area that I've circled. Yeah, quite unfortunate that I can't run the full presentation. So if not, it will be it will be more organized than the way I'm presenting it. So one information comes after the other, but notwithstanding, I will still, pass across the information. So the area that I've circled in green, that is where sample containers comes in. That is where sample container comes in. You will need, so either on-spot analysis or you need to preserve your sample, take them to the lab, for further analysis. Something need to hold your sample, which is sample container. So sample containers required at this stage is required at this particular stage that I've circled, and it is critical to the whole design. So all that I've mentioned above it, everything that comes up below, the effectiveness and the accuracy, is to be determined by the area that I've circled in green. So importance of sample integrity cannot be overemphasized. And achieve it, and this particular area that I've circled can only be achieved onshore, but cannot be changed or modified when you go to when you're on the field. You can't take the wrong sample container from the lab and get to the middle of the ocean and discover that, okay, erroneously you've taken along with you a wrong sample container. It could be costly. So it is important as researchers, as scientists, to ensure that accurate and adequate and proper sample containers are taken along with you right from the laboratory. So sample containers hold your sample prior to analysis, and using the correct sample containers is important to reduce the possibility of contamination of the sample or sample degradation by light, oxidation, or even reduction, and some other process. So there are two types based on the chemical nature. We have the organic and inorganic. So the inorganic ones are your aluminum foil, your glass bottles, and the organic ones, you have the plastic bottles, and then also you have the Ziploc, So those are the organic type. So depending on the physical state of sample, either solid, liquid, or gas, you must ensure that you use appropriate container, basically for safety, and also to maintain sample integrity. So now for liquid, you can use bottle, you can use jar, and then for solid, you can use wrappers, maybe like aluminum foil, or you can use bags. So if samples are to be preserved or fixed, sample containers must be such that it won't react with the added fixers or preservative. For example, concentrated acid will be required for fixing heavy metal samples. So, and also the use of formalin for biological samples too. So these are processes that requires either fixing or preserving the sample. So the choice of your sample container must be such that those additives, those fixers and preservatives will not interfere with your sample or your sample container. Well, will not interfere with your sample container. So they might interact in an irreversible way with your sample, but they must not interfere with the container holding the sample. So also, one of the considerations that could serve as guide is ensuring that the waiting time, that your sample container can sustain the waiting time. So waiting time between the sample collection and sample analysis is very, very important. And sample containers must be able to prevent degradation of sample within this period. So for example, the use of amber colored bottle or amber bottles to prevent photochemical reaction is one of the examples. So once you know that within the period that you will need to wait before the sample collection and the sample analysis, you might possibly expose your sample to maybe sun rays. So it is better, and you know that, and you have the pre-knowledge that photochemical reaction can trigger some other processes that could either claim your analyte of interest or release more of your analyte of interest into your sample. Then you need to look for containers that will prevent that. So your sample container must be designed to allow leak-proof or airtight closure. So you must prevent leakage of any type. Then your sample container must be strong enough to withstand transport and storage. It's very important. It must be designed to withstand transport and storage. You know, in the process of taking your samples from one point to another, you know, something might just happen that, and if your sample container is not strong enough to be able to withstand, you know, these activities, then something, you know, might go wrong along the way. So it must be designed in such a way that it will withstand transport and storage. Then the material or the design must be capable of being tagged and labeled. Some containers you cannot write on them directly. Some, even the tag you put on them, you know, they will easily fall off. So you need to ensure that this is prevented. So the list goes on like that, but the most important consideration is that sample containers must ensure that sample integrity is maintained. Sample containers should be such that will maintain the integrity of your sample. So let's talk briefly about the guiding rule. So depending on the chemical nature of your analyte of interest. So let me talk a bit about the analyte of interest. So if you are given a particular sample, maybe sediment sample. The sediment sample is the sample handed over to you. But what you want to test in it, maybe heavy metals, maybe organics, polychlorinated biphenyls, organochlorine pesticide, that is your analyte of interest. What you want to test in that sample, that is your analyte of interest. So depending on the chemical nature, either organic or inorganic, the guiding rule is you use organic material for samples that your analyte of interest is inorganic. So it's like a vice versa arrangement. So you use organic material to sample or to collect samples whose analyte of interest is inorganic in nature. Let me give an example. So, and the reverse the reverse also applies. You use inorganic material for samples whose analyte of interest is organic. So if organic, now, if your analyte of interest is polychlorinated biphenyls or organochlorine pesticide or oil and grease, then it is expected of you to use inorganic sample containers, e.g. glass bottles or aluminum foil if your sample is solid. So if your sample is solid in nature, then you wrap it with aluminum foil. If your analyte of interest is organic in nature. So let's quickly discuss a possible error, in wrong choices based on this principle. So assuming erroneously, you take along with you plastic bottle to sample for water that is polluted with oil. So what are the possible errors that could set in? The two possible errors that could set in is what I've mentioned here, oil coating and oil retention. So by the time you lower your plastic bottle into the water column, into the water environment, the polluted water that's supposed to go into your container will get that plastic container coated with the oil. The oil that's supposed to go in will get the plastic container coated with oil. And when you get to the laboratory, you want to transfer, for further analysis. As you are emptying your water, into maybe glass containers, in the laboratory, maybe you want to do cleaning, then you will experience what we call oil retention. You won't be able to transfer quantitatively all the oil that is present in the sample that you've collected. You won't be able to and you cannot use organic solvent to start, ensuring that you put into the plastic to ensure that you are compounding the problem. So right from the field, you've introduced a process that will reduce the concentration, of your analyte of interest. So you will have less than it is actually present at the polluted site. So the right container to use, for this purpose will be glass container. So that when you get to the lab, you can transfer quantitatively for further analysis. So as I've said, the reverse is also the case. So if inorganic, for example, maybe your analyte of interest is heavy metals, nutrients. In this process, you use organic sample containers, like plastic bottles, like Ziplocs, like polythene bags, if the sample, if the state of the sample is solid, if the physical state is solid. So let us also look at the possible error that could come in or that could set in if this principle is not actually adhered to or followed. So we're going to look at the sampling for heavy metals. So when you erroneously take along with your glass bottle, this is what will happen. So firstly, let us look at the chemical formation of glass. The chemical, the major component of glass materials, as from our basic knowledge of chemistry, is that it's made of silicate. It's a silica, made of silica. So, and maybe you want to add color, to it, then you need to spike it with salt of different metals. If you want to achieve a brown color, particular salt will be added during the glass blowing. So when this glass material is taken to the field and to be used to sample for heavy metal, to be used to collect sample that your analyte of interest is heavy metal. This is what will happen. You know that during the collection of heavy metal sample, you need to add few drops of concentrated acid. And also know that at low pH, all metals, they tend to be in solution. They tend to be in solution. So what will happen to the inner wall of your glass container is that a process called leaching will start taking place. And once this starts taking place, all the metal that forms your glass, that is contained in your glass, they will start leaching additional concentration of metals into your sample. So you've introduced another source of heavy metal into your sample already at that point. So, and that's setting, error of different types, sampling error, So, okay, my time is fast spent. So, as we all know, ship time is not cheap. Ship time is not cheap. So you need to make sure that the choice of your sample container is gotten correctly even before you leave the lab for the field. So any attempt to take the wrong sample container could be costly. So it is costly to take wrong sample containers. So it compromises the integrity of the sample. It compromises it. So the introduction of error, it also introduced error into the whole research design, as I've mentioned in the diagram that I've shown. Everything that comes before and even after, wrong choice of sample container can crumble the whole process. It can crumble the whole process. And it also cripples the efficiency and the morale of research team. When you get to the middle of the ocean and you suddenly discover that you've taken along with you wrong containers, morals are dropped. Scientists are not encouraged again. So it's either that particular parameter is completely removed out of the list of the parameters to look at. It's not something palatable. It's not something interesting any longer when you know that all that you've been able to put together in the laboratory, get into the field, you are unable to achieve them. So what you sample, so what your sample is sitting next to matters, and it goes a long way to determine the effectiveness of the whole project design. It goes a long way to determine the effectiveness of the whole project design. So I've been able to rush up, and make sure that I run through my my slides within the limited time that I have. So thank you very much. Yeah, I welcome questions, maybe for the remaining few minutes that I have.
Okay, thank you, Nubi. That was great. You managed to get through quite a lot in those 30 minutes.
Yeah.
We still have about 8 minutes left. Does anyone have any questions or comments?
Thank you very much for Olubunmi for your presentation. It was really very thrilling and exciting. I want to find out, so I just want to get a clarification on the kind of sampling material you can use when you are handling organic materials.
Okay. Yeah. Thank you very much. Yeah. As I've mentioned earlier, it is not the matrix that we are actually trying to differentiate in terms of organic and inorganic, but your analyte of interest, what you want to determine in that sample. So you might be given a metric that is organic, but your analyte of interest is inorganic. So it is the principle or the rule that I've given, inorganic for organic, organic for inorganic, is only binding when you've been able to establish your analyte of interest. So now for organic that you've mentioned, I want to believe that you are not referring to organic material, but if your analyte of interest is organic, maybe you want to sample, sample for polychlorinated biphenyls, organochlorine pesticides, dioxin, furans, all those organic, components or organic pollutants in the environment, then your sample container that, is appropriate for that should be glass material. In fact, some, I'm going to definitely, maybe my material will get to you. I provided a link, published by EPA that actually gives a long list of appropriate sample containers, and EPA even suggested a borosilicate glass, which is inert, and is inert to some, chemicals, that are, that are of organic, that are organic in nature. So glass materials, and if your sample is solid, then you will need to use aluminum foil to wrap. But for safety reasons, what scientists do these days is that they will first of all wrap it in an, in an aluminum foil, talking about solid. They can now put it in a plastic, that has seal, for safety reasons. But what your sample is sitting next to matters a lot. I hope I've been able to answer your question.
Okay, but just one more. So if you have, let's say, a wet sample of sediment and you want to analyze for total organic carbon and heavy metals, what would be the ideal container to put this sample in? Okay,
so definitely you are going to use you are going to use different, sample containers. So one will serve, for the inorganic, maybe heavy metals. Then the other will serve for the sample whose your analyte of interest is organic in nature. So you need to separate them, even right from the point of collection.
Okay, thank you very much. Thank you.
Yeah, you're welcome. Yeah. So any other question, or my time is over? Yeah, apology once again for coming up late. Yeah, I sincerely apologize.
Thank you.
Oh, sorry, I'm not actually showing my face. Oh, okay. Yeah, I'm using okay.
All right, great to see you, Nubi.
Thank you very much, Martin. Yeah, I'm happy I was able to make some part of my time.
Absolutely.
Yeah.
I'm going to stop the recording.